The LabScan3D is Luminex's most advanced multiplex analyzer to date. Faster read time, simpler maintenance, accessible to 500 polystyrene beads, providing you a broader combination of analyte detection, and compatibility with assays designed on the LX100 and 200, giving you the flexibility to run classic and next-generation assays. The LabScan3D is useful for the following applications in transplant diagnostics: HLA antibody screening, detection of donor-specific antibodies, profiling complement-binding antibodies, and HLA typing for Class I and Class II alleles. Polystyrene beads are at the heart of all Luminex-based multiplexed assays. The LabScan3D beads are available in a magnetic format, making them easier to handle and automation-friendly. They're internally dyed with precise concentrations of 3 fluorophores, giving each different bead set a unique color code. Using this process, 500 unique bead sets have been created, and biological reagents specific to the analyte being tested can be coupled to the surface of the beads. 2 types of 1 lambda assays can be performed with the LabScan3D: Lab Type for HLA typing and LabScreen for antibody detection. Here's how both of these assays are performed. Detection of HLA antibodies in a test sample can be accomplished by an indirect detection method. For the LabScan product line, an antigen is coupled to the surface of the Luminex beads. When the coupled beads are loaded onto the microtiter plate and mixed with a human serum, the antigen-specific antibody is captured on the surface of the bead. A secondary antibody, labeled with phycoerythrin, is then added to the mix. For the lab-type product line, the beads act as a solid support to immobilize oligonucleotide probes designed to detect specific HLA DNA sequences. Target DNA is amplified from the HLA-specific loci by PCR using biotinylated primer pairs. The resulting biotinylated PCR amplicon products are then incubated with a probe bead array, and the amplicons hybridize to their complementary probe sequence found on specific bead regions. Successfully coupled bead probe combinations with target DNA are incubated with streptavidin phycoerythrin, which binds to the biotin on the DNA. Once the samples are ready, they can be read by the LabScan3D system. A sample probe draws the suspended bead with a captured and labeled analyte into the instrument. The beads are carried by the system sheath fluid into a cuvette, where the randomly distributed beads line up in a single file. This allows the particles to be measured discreetly. As each bead passes through the cuvette, a red laser excites the internal dyes, allowing proper classification of the bead, which, in turn, identifies the analyte attached to the bead. A green laser excites the labeling molecule bound to the analyte on the bead, providing information on the amount of analyte present. Both classification and reporter readings are made on each individual bead, so the LabScan3D system is able to precisely determine multiplexed assay results. Luminex Exponent software processes the data and exports it to 1 Lambda's proprietary analysis software, HLA Fusion. The LabScan3D system assures high throughput without compromising flexibility or performance and can help optimize your workflow to meet the constant demands for high-quality data, with features like faster read times driven by dual sample fluidic paths and an increased sample injection rate, simpler maintenance with automated commands for probe height adjustments, startup shutdown routines, and enhanced calibration. Beyond the mechanics of the LabScan3D, the state-of-the-art exponent acquisition software adds a whole new element of freedom by providing remote web monitoring, more walkaway commands, and set key function alerts via email. To accommodate all the progress made with Luminex technology, the system is fully compatible with the current lab type and lab screen assays, along with our future next-generation assays with extended analyte capabilities made possible with the availability of 500 polystyrene beads, exclusive to the LABScan3D.